Welcome back to Human Biology with Dr. B. Today we're talking about chromosomes and cell division. So, what is a chromosome? You hear all this talk about chromosomes and how they carry genes and how they're all this genetic material. Well, the basic definition of a chromosome is the DNA wrapped around histone proteins. As you can see, all the way here in the nucleus, we have chromosomes, and when they are in metaphase, which we'll talk about in a moment, you can very clearly see what we call the sister chromatids. Also, we'll talk about that in a moment as well. Chromatids are the fully condensed version of a chromosome. And here you can see that they are supercoiled again, wound around, and all the way down here you can see what we call the nucleosome. The nucleosome is two circles of DNA wrapped around 4 histone proteins. And the histone proteins act like a spool, and the chromosome wraps around it so that it can tightly pack in. And then these spools of thread are then wound and packed more tightly together, and then coiled some more, coiling back on each other until they have fully condensed into sister chromatids. Now, in a non-dividing cell, the chromosomes just look like this. The chromatin is completely relaxed. It's not a big deal. It's whatever. Remember how I talked about that pot of spaghetti? So you boil your spaghetti, and then it's done. And so you pour it out into a colander. Now picture this with me. Dumping the spaghetti out. Poof, there it is. And you have all of these spaghettis going everywhere. Now, if you were to, gross, you weren't raised in a barn, so you're never going to do this. But if you were to reach in and grab a handful and just pull it out, some of the spaghetti strands would break. And so in a non-dividing cell, the chromatin is completely unwound. So if we were to go through cell division and just pull the DNA to either side, the DNA would break and we would have mutations in the cell, and we don't want that. So we compact them into chromosomes and sister chromatids. There's a lot of talk about a gene. Now, a gene is not only the denim that you have on your body, which is spelled G-E-A-N, but a gene, referring to biology, is the functional unit of heredity. Now, within a piece of chromosome, as you can see here, there is this massive amount of DNA, and only certain pieces of DNA legitimately code for a function. Now, a gene is anything, a specific segment of DNA that can provide a functional protein. It codes for the synthesis of a protein. And this goes all the way back to the central dogma of biology, DNA to RNA to protein. And then we get to talk about the difference between somatic cells and non-somatic cells, or what we call gametes. Now, somatic cells is all the cells in your body with the exception of your gametes. And the gametes are only 2 different types of cells, sperm or eggs, and you either have one or the other, the end. Now, somatic cells have all 46 chromosomes. Now, you get 23 from your mom and 23 from your dad to make a total of 46. And we call this diploid, that you have you have one pair of each chromosome. You have chromosome 1 from your mom and chromosome 1 for your dad. Chromosome 2 from your mom and chromosome 2 from your dad. And we call this diploid. And this is denoted 2N. Now, sex cells or gametes, sex cells can have one of 2 different types of chromosomes. They can either have an X or a Y. Now, sex cells, gametes, are haploid. They only have one set of chromosomes. And this one set of chromosomes is only 23, and they can come from your mom or your dad or anywhere. And we'll talk more about this variation in a moment. But we refer to 1N as haploid. And all of our gametes are haploid. They only have one set of the chromosomes. And this is the alternation of generation for humans. So what does alternation of generations mean? It means the difference in when the organism is haploid and then diploid. And so this is what a human alternation of generations look like. Also, this baby is exciting. Okay, so We spend the majority of our time as a diploid organism, fully formed diploid organism. And it's only through the process of meiosis that these cells, the sex cells, become haploid. And then when these haploid cells join, which we call fertilization, they form a zygote, which in turn turns into a baby, which turns into an adult. Which brings us to the cell cycle. So what is a cell cycle? It's the cycle that a cell goes through. Duh! It's the sequence of events from a cell's origin through its own division. So remember how we talked about all living things go through growth and repair? That's one of the main things, qualifications to make something living. Well, the way that cells grow and repair is through mitosis. There are two major phases. Interphase, which has 3 subphases, and then cell division, which basically has 2 and then 5 subphases underneath that. So this is what we call the cell clock. Here you can see that the majority of the time the cell spends in interphase, which we will talk about more in depth in just a moment. And only a small portion of the cell's actual life cycle is spent in mitosis. So here you can see we have the cell, again, back to the chromosome, and you can see how the chromosomes interact with the cell cycle. So when the cell is in interphase, the chromosomes are unduplicated, and they're just that big pot of spaghetti, unwound, no big deal. Now, once the chromosomes become duplicated, this occurs in interphase, we'll talk about, and then they begin to condense, wind back around the histones, supercoiling, and then fully condensed into what we call sister chromatids. And here you can see this is not to scale. It shows that interphase is a small portion of the cell cycle and mitosis takes a while. Now, mitosis does take a while, but it is a very small fraction of the actual cell cycle. So what happens in interphase? There's 3 basic parts. There's G1, which is the cell grows, duplicates the organelles. So the organelles duplicate first, and then S, the S phase stands for synthesis, where DNA is then synthesized. Now the 2 chromosomes that are copied remain attached at what we call a centromere. And once they are condensed, they are called chromatids, sister chromatids. And they're not condensed until into mitosis. And then G0 is where cells that don't divide. This is what we call senescence. They basically carry out normal functions. It's G0 where the cells carry out normal functions. Like I'm just being a nerve cell today. That's what I'm doing. Not dividing. And then we get into mitosis. Now we have 4 stages plus cytokinesis for a total of 5 stages of mitosis. And I remember them because we go interphase, prophase, metaphase, anaphase, telophase. And if you look at the letters, they form IPMAT, which for Colorado, I use the mnemonic device in Pueblo mice are tall. IPMAT. So let's look at what happens. So in prophase, and you do need to be responsible for what happens in each phase. In prophase, the chromosomes begin to condense. The DNA wraps tightly around the histones. The nuclear membrane begins to break down because remember, we're going to need to pull the DNA, the chromosomes to either side of the cell in order to make 2 cells. So we need to remove the nuclear membrane, and the mitotic spindle begins to form. And this, remember, from our previous section of the cell, the spindle is 2 basic organelles, the centriole and then the microtubules that act like a fishing line. They basically attach to the chromosomes and then reel them into either side. The duplicated centrioles begin to move to the poles and attach to the centrosome. Then in metaphase, the main way that we know metaphase, so we have prophase, metaphase. Metaphase, the condensed chromosomes line up along what we call the metaphase plate. It's very obvious. And this is a function of the mitotic spindle pushing and pulling the chromosomes so they line up. And then anaphase, animation, ana, that's what that means at the beginning. It means movement. The chromosomes begin to pull to either side of the cell. And then telophase, the nuclear membrane begins to reform, a cleavage furrow between the cells so that it pinches the cells off. And then cytokinesis is what we refer to as cytoplasmic division, where the 2 cells finish dividing and the organelles move to either side, and voila, we have 2 cells. And so this is what it physically looks like. So remember, in interphase, we're duplicating our chromosomes. That happens in the synthesis phase. And then in prophase, the nuclear membrane breaks down, the chromosomes condense, the mitotic spindle begins to form and move to either pole. Again, the mitotic spindle attaches to each of the sister chromatids. They're called chromosomes because they're still connected. In metaphase, all the chromosomes due to the mitotic spindle are pushed and pulled, and they line up along the metaphase plate. This is what this looks like. And then in anaphase, they begin to pull to either side so that each cell, look at these micrographs, it's beautiful, will receive one copy of each chromosome. And then in telophase, the nuclear membrane begins to reform. The mitotic spindle goes away, and we begin to see what's called a cleavage furrow. And in cytokinesis, it's full division of the cytoplasm, and then we have 2 new cells. The DNA begins to decondense, the nuclear membrane begins to reform, and we have 2 new cells at the end of it. I will post this video on your learning management system. And then we get to meiosis. Now, mitosis is cell division. Meiosis is the creation of gametes. So the whole purpose of meiosis is to half the number of chromosomes, because every cell in your body has 46 chromosomes, except for sperm and eggs, which have 23. So the whole purpose, introduce variation, cut the chromosomes by half. So meiosis is the number one way, or the only way, that we are able to keep the number of chromosomes consistent from generation to generation. So that your parents have 46 chromosomes in all their somatic cells, and you have 46 chromosomes, and then your kids, should you choose to have them, will have 46 chromosomes as well. So the way that variation is introduced in this is by 2, there's more than 2, but for your purposes, 2, independent assortment and crossing over. And meiosis, unlike mitosis, has 2 divisions. It goes through meiosis 1 and then meiosis 2. So in prophase 1 of meiosis 1, because we go through prophase, metaphase, anaphase, telophase in meiosis 1, and then we go through it again in meiosis 2 because we need 2 divisions. So prophase 1 is where synapsis occurs. Synapsis is right here, where non-sister chromatids line up. So the difference between a sister and a non-sister. Sisters are copies of each other. Non-sisters are the one from your mom and the one from your dad, which is why one is pink and one is blue. So these chromosomes from your mom and your dad, as I mentioned in class, let's say this chromosome has hair color, eye color, and skin color. So this chromosome from your dad will have the same genes. It will also code for hair color, skin color, eye color. Although those aren't on the same chromosome, it's just an example. So these are what we call homologous chromosomes. They have the same genes coded on them. However, a sister chromatid is an exact copy of one, and they're attached right here at the centromere. So in prophase 1, we have crossing over, where non-sister chromatids, homologous chromatids, they basically line up and they exchange genetic material. And this allows for the first introduction of variation. Metaphase 1, it's very important that you note the difference between metaphase 1 and metaphase 2. So in metaphase 1, homologous chromosomes line up on the metaphase plate. So what does this mean? That means dad and mom can go to either side of the cell. So think about you. You have genes from your mom and from your dad. And when in you gametes are made, the chromosomes that you receive from your mom and the chromosomes that you receive from your dad can get pushed and pulled to either side of the gamete. So that way, your kids don't necessarily just look like you or just look like your mom or dad. They can look like a whole host of things, which is amazing, the amount of variation this can introduce. So metaphase 1, the first introduction of variation, is what we call independent assortment. So they pull the homologous chromosomes randomly to either side of the cell. And then anaphase 1 is where they begin to move, and each chromosome still has sister chromatids. However, at the end of anaphase 1 is where we say these cells are now haploid. And the reason why is because they only have one copy of each homologous pair. So telophase, again, Nuclear membrane reforms, cytokinesis. Now we have 2 cells. Interkinesis is the brief resting period. I believe it's in this state that all of the eggs are stored in the female, and then when she reaches puberty, will go through meiosis 2 and ovulation, etc. So here you go. So prophase 1, we break down the nuclear membrane, the spindle. The spindle forms and moves to either pole. It attaches to the chromosomes. Non-sister chromatids, homologous chromosomes, cross over. We have the introduction of our first variation. Then in metaphase 1, we line up. And then anaphase 2, this is independent assortment, where mom and dad can randomly assort to either side of the cell. Telophase 1 and cytokinesis reform the nuclear membrane. Spindle goes away. And all of a sudden we have random distribution of your mom and your dad's chromosomes in either side. Then we get through meiosis 2. Prophase 2, the chromosomes do not cross over, and the reason why is because except for the crossing over that happened in prophase 1, these are the same duplicated chromosomes. They're still sister chromatids. Metaphase 2, they line up. Anaphase is where the sister chromatids separate, and we have the third level of variation, where some of the chromosomes have crossed over and some haven't, and we have independent assortment and law of segregation. And this is the law of segregation, where they segregate to either side of the cell. And then we have telophase and cytokinesis, same thing. And here's what it looks like. Prophase 2, nuclear membrane breaks down. Do you see a pattern here? The centromere sends out microtubules. It attaches to the centromere. These get pulled and pushed to the metaphase plate. The sister chromatids separate, and all of a sudden we have 4 haploid cells. Now, in males, 1 diploid cell will result in 4 haploid sperm. However, in an egg, because the eggs are one of the only cells in the human body that you can see without a microscope, because it's that big. And the reason why it's that big is because 1 diploid cell results in 1 haploid cell in 3 polar bodies. Now, these polar bodies get none of the cytoplasm. So the cytoplasm continues to duplicate until we have this big ovum. And these 3 polar bodies, all they have is the genetic material, and that gets resorbed, bye-bye, whatever. And this is so that the egg gets all the nutrients so that when it's fertilized, it can become a baby. Okay, and this is what happens. So here you can see, this is how much smaller the sperm is compared to the egg. And you have all 3 polar bodies and 1 egg. This is the 4th level of variation in meiosis, because we don't know which one of these variations will become the actual egg, and we also don't know which one of these sperm will eventually fertilize the egg. So now you should be able to do these 5 things. Describe the difference between a chromosome and a chromatid and the different levels of compacting. Apply the definitions of haploid and diploid. Describe the human alteration of generations. And list in detail the steps of both mitosis and meiosis. And then I need you to be able to describe how both meiosis and mitosis are the same and also different. Thanks for joining me. It's been a great time on Human Biology with Dr. B.